based off of what they look like. We got the line, the sign, the LTR, and the DNA elements. Fun, right? It's almost alphabetical, but not really. At least the line and sign are in alphabetical order. So that's somewhat helpful. As you can see, they are ranked or listed in an order of abundance. From most abundant to least abundant. Now beyond that, there are some important considerations to think about. So as we go forward, we're going to break them down based off of not so much what they do, but how they move. And the DNA element, what we call DNA transposon, has a different mechanism of movement than the others do. That'll be interesting as far as how we look at these things. The slide, or at least the table from the textbook that is the reference for most of the things I talked about, is just shown here to give you an idea of overall numbers. You don't need to worry about the actual percentages, but they're sometimes fun to think about. And what's also nice about this, it lists some of the, actually tells you how many there are, not just how much of the genome is actually this thing, but how much of it is one thing. But 20% is kind of abstract. When you think there's almost 870,000 copies of this one group of transposons in you, or in every single cell of your body, are 868,000 copies of this one little thing. Is that frightening, astounding, interesting, boring? Is it boring? Let me know if it's boring, because I'll keep going. I'll try to bore you to death. And I can do it too. Now what's fun about this slide also is, based on percentages, this is the most abundant, but based on total numbers, this is the most abundant. So what do you think there is different between this one and this one? Anyone want to take a guess? How can you have less number, but be more of the overall average? Or bigger. And that's what the L is going to stand for as you get onto the next couple slides. L is for long, S is for short. But still, think about nearly 1.6 million copies plus 870,000 copies. Do that math really quickly. I don't know what it is. That's a lot. There's a lot of these in our genome. Why are there so many? And why are they still there? The line and sign and the LTR are a group of transposons that work in a similar way. What makes them different is these two are similar, this one's different, but all three are together in one little way. Then we have the lowly one of the bunch, the DNA transposons. Then there's some that we don't really know what they are because they're different than these four. But still, overall, half of the DNA inside every cell is not useful. Think of how much better we could be if we got rid of all that junk. Or think of what we might look like if they got rid of this DNA that's been in our genomes for several thousand million years, whatever you want to think about evolutionarily speaking. Remember, a transposon is a movable piece of DNA. It facilitates its own movement from one position to another. So as a general idea, if you think about a circular piece of DNA, like in a prokaryotic chromosome, a transposon might be in this position to start with. If it's active, it's able to cut itself out and move itself to a new position. And so maybe it will cut itself out and move here, or there, or here, or it could go back fairly close to where it actually was. The transposon cutting itself out and moving it is a random process. For scumpit, you never know what you're going to get. Also sort of applies here. You never know where this transposon is going to go. So when we think about genes and transposons and their effects, remember we have this basic idea of how DNA is organized and how the gene is organized on the DNA. our gene, we have our promoter, and we have our terminator. And as we talked about briefly last time at the end of the class, if the transposon were to move and jump in the middle of the gene sequence, that typically makes it non-functional. Protein doesn't work anymore, and that's the case we get with the green versus wrinkled seed idea that we talked about earlier on this semester. So transposons can move and they can turn, they can inactivate a gene, basically what's called a knockout. So it turns it off, makes it dead or non-functional anymore. What if it gets into the promoter area? How does that affect?
It will start transcription.
Right, probably there's no gene expression whatsoever. Or if it gets in the terminator, and you're still transcribing, but where do you stop? So who knows? Maybe you make an RNA too long or something. If it gets in the middle, maybe that also sort of makes things wonky also. And so when we think about where transposons move, the likelihood of hitting a gene is actually pretty small. But hitting a gene is going to have not always, well, it's going to have a detrimental effect, but it can also go somewhere else and also have a detrimental effect. Which is why it's important to think about these things are non-functional right now in our genomes. They've basically been dead for however long evolutionary history says. I think it's now 1.5 million years. That's before, that's pre-human, but in the eukaryotic cell line, they believe these transposons stopped working very early on, or at least long time ago. Every transposon typically encodes its own enzyme called a transposase. And this transposase will be transposon specific. The transposase for the line will only work on line and not on site. For example, you have to work on your own type of transposon. And generally what they do is they recognize specific DNA sequences that are at the end of every transposon, which can either be a direct repeat or an inverted repeat. And so if the word sequence is in the same orientation, we call it a direct repeat because it's going the same way. But it's not sequence, it's ATGCCC, whatever. And it's the same here, it's a direct, if it's going in the opposite direction, we call that inverted repeat. And so if it was ATGC over there, it'd be the opposite of that, which is GCTA, or however it's opposite going in the other direction. Either way. But these are basically the bookends. Every transposon has a bookend, or two, that tells us this is the end of the transposon, and the transposase enzyme is going to cut the DNA on either side of the transposon and move it to another location. So it's going to cut DNA actually twice. Once in the original position, and a second time in the new position. Because that's to cut the DNA, the transposon out, and cut the DNA to where it's going to go in. So imagine here, imagine the transposon was in there, we cut it out, or at least cut DNA and insert the transposon, and it's there. But where did it come from and how to cut itself out in the first place? The exact mechanism is not important. But for a DNA transposon, because it cuts itself out and then moves itself to a new location, it limits how many copies of this transposon are actually present. And one of the things I want you to think about with the DNA transposons, the number of transposons never increases because of the transposition process. It cuts itself out, it moves it to a new place, so it's only ever in one place at a time. Which limits how many of these things are actually available, because every time you move, you don't get a new transposon, you just get it in a new place. But you don't have to cut the DNA. Where the transposon is, to remove it and cut it in a new position where we put it. That makes the transposase a double-stranded DNA cutting enzyme, because that's the both strands of DNA molecule. That's something that's going to be more important as we go forward this semester. Does this cut only one strand, or does it cut both strands? And for transposase, it definitely has to cut both strands of the DNA, because the transposon is a double-stranded DNA molecule. It's in the DNA. Everything in DNA is a double-stranded thing. We cut it out, move it, it moves itself. Fairly straightforward, right? It's like doing any type of transplantation. Cut it out of somebody or something, put it into somebody or something else. Whether it's heart surgery or ligament surgery or whatever. You're taking something from one place and putting it into a new place. You're not making a new one. You're not copying it. Say, here, you make a copy of you so I can give it to somebody else. Doesn't work that way. And sort of got ahead of myself as far as the slides go, but if a transposon moves itself into the gene sequence, between the start codon and the stop codon, that typically is going to make a non-functional protein. We get a loss of function. So that is what we call an gene inactivation or protein inactivation, depending on how you think about things. But getting a non-functional protein is what will typically happen when a transposon jumps into a gene. But if it jumps somewhere else, maybe it jumps into a motor, which could have effects on whether this thing is expressed to the same level or to a different level. And most likely, if the transposon gets into the promoter, it probably breaks the promoter. So you get a decrease in gene expression. And we call this hypomorphic. Does anybody remember talking about hypotonic and hypertonic solutions? And before it's the same idea, hypotonic means low, so we have a lower expression. We're going to talk more about hypo and also hypermorphic effects as we go forward this semester. The transposons can be pretty good mutagens. It's one of the things on the list. If you go back to the slides, if DNA pieces move themselves to new locations, that can cause negative effects for things. So the DNA transposon, what we call the DNA element, cuts itself out and moves it to a new location. For the other three, we call them retrotransposons. Does anybody know the most famous retro? And this is probably pre-history for everybody. There was a virus in the 90s that was supposed to kill everybody. They call it HIV. Does anybody know anything about HIV? They call it a retrovirus. Does anybody know why? HIV is an RNA virus, which means to make copies of itself, you have to go reverse transcription, go from RNA to DNA, and then make more copies of DNA, and make more copies of RNA, and then you can make more viruses. And that's fairly the reason why these are called retrotransposons, because there's an RNA molecule that's going to exist in this. We call this, basically I call it an RNA intermediate. The transposon itself is a DNA sequence, because it's in the genome, so it has to be DNA. Instead of cutting the transposon out, you make a copy of it. And that copy is an RNA copy. But the RNA copy has to be made into DNA before it can be inserted into a new location. So transposition is fairly similar. Take this thing and put it into a new location. What makes the DNA transposons different from the retrotransposons is that RNA intermediate, and the fact we don't cut out the original copy. It stays there, its original position, and we put a new one to somewhere else. So now instead of having one copy of transposon, we have two. And 2 makes 4, and 4 makes 8, and 8 makes 16, and then 32, then 64, and 48, until you get to 1.6 million or 870,000. So when you think about what retrotransposons do, it kind of makes sense. There's a lot of them in the genome because every time they move, they multiply. They never get cut out. They just make a copy. That copy gets put somewhere else. And the exact mechanism is not important. But think about, there is an RNA intermediate in the transposition process of retrotransposons. And we don't cut out the original piece, but we leave it there and make a new one somewhere else. So we do get basically an increase in the number of these transposons every time they decide to move. The LTR transposons are what we call LTR. The line and the sign are called non-LTR. LTR stands for long terminal repeats. Whoop-de-doo, as they say. They don't really care. But all transposons have repeats at the end. This one just has really long repeats, which makes it its own sort of unique type of transposon. And then in the non-LTR, the line and the sign, don't have these long terminal repeats. What makes one different than the others are long versus short. Now you might be asked to tell me what these acronyms stand for at some point or another, but maybe not. Maybe it's all just simply an abundance question, and which one of these transposed using an RNA intermediate. The transposons are really powerful, at least in the laboratory setting. We can engineer them to move things we want to move to a new location, or put this somewhere unique, or into a new organism. So they have very powerful tools that we can harness the transposition process. We can make them transpose whatever DNA we want them to transpose. And so gene therapy is where transposition might become very useful. Well, it hasn't really worked out. We would like to. Imagine you take repeats and put a good copy of a gene that someone has a mutation in. That mutation is causing a disease. So let's try to replace that missing function by putting a gene and transposon, inserting that into a human, say, inject that DNA in there, and now you're cured. In practice, it sounds good, but in practicality, gene therapy has some issues. So you may want to try to think about why gene therapy might have some potential issues. How many cells are there in the human body? How many cells does gene therapy actually work on? That's a big problem with the idea of gene therapy, is how do you get to every single cell in your body? Or does it only need to go to some cell, because only those cells use that to code DNA? But still, you have to get enough in there to be useful, and that's one of the problems with gene therapy. Are you getting enough coverage? There's one more cool thing about gene therapy that will come up later this semester. That's a sort of a twist to this idea. That's one of the things we're trying to do to help with treatment of disease, is replace bad or non-functioning pieces of DNA with good functioning pieces of DNA. So we got a lot of these transposons in us. So our genome is all of our pieces of DNA. Does anybody know how many pieces of DNA are in the human genome? Everybody should know this, like basic fact of humans. We have this many chromosomes. No, we have 46. Remember, we are diploid organisms. So we don't just have 23, we have 2 copies of everything. So there's 46 total chromosomes, 23 from one parent, 23 from your other parent. So we have 2 copies of everything. But our genome is all 46 of those chromosomes. Remember, eukaryotic chromosomes are linear. We'll talk about telomeres and what they look like here at the end of a chromosome in a second. Prokaryotic organisms, there's a circular chromosome. There's also another small piece of DNA that we can find in prokaryotic organisms fairly often. This is what's called a plasmid. To give you just a size perspective, a average prokaryotic chromosome is about 5 million, big difference. 5 million base pairs. A plasmid might be 5,000 to 10,000. There's a pretty big difference in size there, but it's still just a small piece of DNA. So genome is all of your DNA. Mostly it's chromosomes, but for some organisms, it's chromosome plus other additional pieces of DNA. For humans, the 46 chromosomes are not the only thing that makes up our genome. There are other things inside a cell that contain DNA. Did they talk about this in 140 or no? Besides the nucleus, where else can we find DNA inside a human cell? And mitochondria has its own DNA. We have to account for that. That is part of our genome. Even though it's not part of our 46 total chromosomes, it's still DNA inside our cells. So it is part of our total genome, and it's typically forgotten. And for plants, chloroplasts have their own DNA, so it's nucleus, chloroplast, and mitochondria. They all have to get added together to make the genome of those individuals. As far as size goes, we do see differences in how much DNA does an organism actually contain. And we also see differences in the organization of that genetic information. Don't memorize the numbers, but the numbers are there just for size reference. Again, the smallest genomes are in the viruses. The biggest genomes are in the eukaryotic organisms. The prokaryotic organisms somewhere in the middle. And for cells, the DNA, the genome is DNA, and that's all it is. Just a question of, do we have circular chromosomes or linear chromosomes? I should probably update this. The DNA inside the mitochondria is actually circular, but I'm not going to ask you that. That's just an extra little bit. But for viruses, we do see a wide variety of organisms out there. And it is thought that the diversity that is seen in viruses is more than 10 times greater than the diversity we see with cells. There are a lot of different viruses out there. It's not just, are they DNA or RNA, or are they linear, or are they circular? At some point or another, scientists wondered, well, what is the least you need to actually exist? And so somebody actually tried it. His name was Craig Bittner. He took the smallest known organism, which is called mycoplasma. That's not something you need to remember. But he used something that already had a small genome to start with. And he kept mutating genes until he found something he could get rid of. And he basically made an artificial microorganism where you could get rid of about half of all the genes that are present there. It still works. It only works under one condition, the condition he was growing it in the lab. Doesn't mean it'd be successful out in the real world, but when you think about it, that's pretty cool that you can get rid of half your genes and still actually live, albeit in a weird situation. What I think is fun is that when you look at those 473 genes, about half of them are involved in copying DNA and making proteins. So cell division is really important, and making proteins is really important. Everything else, you just need a little bit of stuff. There's one other fun sort of fact about Craig Bittner's experiment, and that's this category of things. Genes that we don't know what they do, but are essential for life. There are a number of genes in the human genome that we have no idea what they do or what function or purpose they serve. You can make yourself famous by identifying what these things are and what they do and why they're important. But if it was easy, it would have been done already. But you're smart. You're the future. You can do it. I have faith that someday we will know what the unknown actually is. But think about that. I mean, does that surprise anybody that there's things out there where we don't actually know what they do yet? A lot of people sort of assume we know everything in science. We don't know everything in science. So Craig Bittner interested in looking at what is the minimal genome for a microorganism. Generally speaking, we associate complexity with complexity. complexity. I know that's sort of a weird thing to think, but complexity in the first place doesn't mean complexity in the second place. Would you assume that a snail is more complex than a human? Why not? One is bigger, a lot of extra features, one is much smaller and much simpler. So why isn't the snail just simpler than a human? Is it? Is it not? Maybe you sort of see the trap I'm trying to lay for you. Size is not necessarily an idea of complexity. And so just because you're bigger doesn't mean you're more complex. There is a certain point where complexity and size do sort of matter, and it matters on a large scale. For viruses, they have less DNA and they're typically simpler. They can't live on their own. Bacteria are much smaller than a eukaryotic cell. They have less DNA, mostly because of size constraints. The size of a nucleus is kind of the same size as a prokaryotic cell, sort of for reference. Eukaryotic cells are just bigger, not because they have more DNA, but because they have more extra things, like mitochondria and all those other things. And we also sort of associate its complexity related to the number of genes that an organism has. And the answer is generally no. We don't really see a true correlation that we can say that, yes, this small genome means you have more genes because you have more space. But I do want to highlight one basic fact. Prokaryotic organisms have a very small set of genes. This is a eukaryotic organism, has a very similar set of genes. But the size of a yeast cell and the size of a bacterium are kind of the same. So both single-celled organisms. So for single-celled organisms, that's all you need. When you get into multicellular things, I mean, nematodes, you ever seen a nematode before? It's like, I wouldn't say that it's a microscopic worm, but it's really small. It's the scourge of the agricultural community. But it has 20,000 genes, and we only have 25,000. Right. So, are we more complex than a little bitty worm that's that big? I mean, look at this. A Arabidopsis, that's a plant. A plant is more complex than humans are. If you look at the number of genes, because they have 28,000 genes. What's similar between these three here is the size of the genome, and it's on the average of about 100 to 200 compared to 2,000 to 3,000. Differences in the amount of DNA, the same number of genes. Why is this important? We talked about this already, gene density. Prokaryotic organisms, like bacteria, are really good at using the DNA they have to put genes in all the places that they have available to them. As you get more and more complex, you get less and less good at using your DNA. You get more DNA, but not as many genes. So there's a disconnect in genome size and gene number. So we can't really say that there was a correlation. Yes, more DNA means more genes are possible, but there's a problem. And most of that problem is transposons, introns, take up a lot of our space. But we don't see all that same stuff in single-cell eukaryotic and single-cell prokaryotic organisms. So size does not necessarily mean anything as far as gene number or anything else. But there is some basic rules. That's just what comes to gene density. So make sure we understand gene density. I'm not sure why I put the same slide in here, guys. It's not really the same slide, but still. I mean, genome size, not correlated with gene number. Not correlated with gene number. I just really want to stress that one idea apparently. No correlation between genome size and gene number. There is a correlation with gene density. I know this is pretty boring stuff. Facts are always pretty boring. So it does lay a good foundation for what we can talk about eventually, which is how can we use these differences and more, some more of these differences that we can look at. So the basic part of that first set of slides was to get us an idea of what is in a chromosome. Now we can talk about, well, what is a chromosome? What does it look like, and how can we tell how many chromosomes are in a cell? Then we'll go back to DNA to talk about some individual ways to talk about DNA differences between one individual and the other. We've already seen this slide. It's not a new slide. You should already know about Meischer basically finding DNA. What's important about the slide to remind you is that chromosomes contain DNA and proteins. We mainly focus on the DNA for a lot, but we do have to talk about the proteins quite a bit in this section here. This should also not be, this is not a new slide, it's a repeat from the previous section, just to remind you. A chromosome is a basic unit of DNA inside a cell. Eukaryotic organisms always have multiple chromosomes, and those chromosomes are almost exclusively linear in appearance. With the telomere at one end, the telomere at the other, and somewhere between those two places is another place we call a centromere. And as I mentioned before, the centromere is important for cell division and the ability to move that DNA around. Telomeres are protective devices. We'll talk more about how they protect and what they protect as we go forward, and we very quickly, we should give the answer. But it turns out that every chromosome contains two different types of DNA. We call them euchromatin and heterochromatin. EU basically means true. The true chromatin versus the different chromatin? I guess hetero, different? I don't know. So basically, when we draw a chromosome, we can divide the chromosome into different parts. Heterochromatin part and euchromatin part. And so if you think about a basic picture of a chromosome, this would be sort of a working format. Telomeres at each end. Centromere, as we'll see in a minute, centromere is not always in the actual center. Then we have what's in between. What's in between is what we call euchromatin. The telomeres, the centromeres are what we call heterochromatin. And the basic idea that we're going to see going forward will be on the next slide. I'm just sort of getting ahead of myself. Euchromatin is where most of the genes are found. Very few genes are found in the telomeric area or the centromere area. There are a few, it's kind of rare, but for the most part, we think about euchromatin, that's our protein coding information. Now it doesn't matter, but they identified these different types by staining methods. And so they found that one thing stained more quickly and the other thing didn't, but that's not really important for us. What is important for us is that we can differentiate the chromosome into different parts. A euchromatin area and a heterochromatin area. And generally speaking, there's going to be 3 heterochromatin areas and only 2 euchromatin areas. Here you see 3 of these yellow-orange pieces, that's the heterochromatin area, and 2 of these purple pieces, that's the euchromatin area. We do see a little bit of variation in where the centromere is, so there is going to be some differences between one chromosome and the next. But again, the euchromatin is where we find the highest abundance of our genes that are located on our chromosomes. Very few genes found in the heterochromatin areas. What we do find a lot in the heterochromatin area are these repeat sequences. And the repeats in the telomere are different than the repeats that are in the centromere area. Because If they were the same, they'd be called the same name. To make a protein with one function, you need some variety. You can't use the same letters over and over again because proteins that have the same amino acid typically don't do very well. So with repeat DNA, there's very little variety in there, which is why there's not many genes located in the heterochromosome area. But luckily, because there is the centromere area and the telomere area, any problems or changes that occur in the centromere or the telomere are not going to affect proteins or protein functionality or gene expression at all. Because it's, who cares DNA? And that's one of the things we can think about. Introns and heterochromatin are non-protein coding DNA sequences. So are they important? Not for gene expression, but for other purposes. They do have an actual purpose. And we do again want to think about what that purpose is as we go forward. We'll start with the centromere. A lot of people think the dot is the actual centromere, but it's not just the dot. That's kind of a misconception. As you can see, the centromere is a fairly long region of DNA right there at the middle. Every chromosome is broken into two parts by the centromere. We call a P arm and a Q arm. The P arm is always the shortest part of the chromosome. The Q arm is always the longest part of the chromosome. And so if you move that centromere closer to one side or the other, P gets smaller, Q gets bigger. On this slide, there is an important thing that's typically forgotten quite a bit, is the DNA sequence that we find at the centromere is what we call an alpha satellite DNA. It's the name of the repeat DNA that's commonly found at the centromere. So why do they call it centromeric repeat? I don't know, but they call it the alpha satellites or alpha satellite sequences. But that's what your centromere is made of. Repeat DNA called alpha satellites. And again, the centromere is important. It's a place where we hook on proteins to move our DNA around during mitosis and meiosis, so that cell division works properly. Based off of where the centromere is located, we classify chromosomes in one of three different groups. And generally speaking, the metacentric chromosomes have a P arm and a Q arm that are more or less the same length. So our centromere is right in the middle from one end to the other. Acrocentric is when the centromere is located very near to one end, so you get a very short P arm and a really, really long Q arm. And you have the one that's kind of in the middle of the middle, the sub-metacentric. And these are just numbers for illustrations. Sometimes I don't use these exact numbers, but it's plus or minus 5, or plus or minus 10 is close enough to hopefully you can understand. That if it's 80-20, maybe you could argue it's this, but if it's 95-5 or 85-15, you should think, yeah, that sounds like an acrocentric. Do you know what acrophobia is? Acrophobia? No, it's not fear of gymnastics, it is fear of heights though. That's why it's called acrocentric, because it's on the end. It's sort of the high side of the chromosome. The only thing that the centromere position matters for is for talking about the DNA. Otherwise, it really doesn't have any impact whatsoever on how the chromosome actually functions. But scientists like to name things and give things locations. And it's easy to talk about where something is based off of where it actually is. So a lot of times we'll see things like 11p1-3. Tells you the chromosome number, if it's on the P arm or the Q arm, and how far away it is from the centromere. Sort of a general idea to think about. But I'm going to ask you to locate things. When you look at the DNA when it is moving around, it does have a unique shape. So we do see that chromosomes do look differently when they're being moved around, but that's just a fun little fact, not something you need to remember. Again, the centromere is where we attach these proteins that are going to be needed for cell division. So here's an illustration of all of the chromosomes that we find in humans. They're ranged from biggest to smallest. 1 through 22 are what we call the autosome chromosomes, the non-sex chromosomes. We have the X chromosome and the Y chromosome down here at the bottom. Hopefully you can kind of notice this little dot in the middle of all of them. It's sort of highlighted by this line. That's the centromere, and the other end is the telomeres. So you see there's a lot of variety that we have out there. And I'm going to ask you to look at this on the side. This is a submetacentric or an acrocentric, but I would call this a submetacentric. This one right here, these right here are probably acrocentric because there's very little P arm to begin with. Some of them are meta, you know, you get pretty close here to the middle. Number 1 is pretty close to the middle. Number 3 is pretty close to the middle. But number 4 is more to one side relative to the other. And what I don't remember is what is this letter for. It does have some meaning, I don't remember what it's for, so it doesn't matter, but isn't it neat, colorful, fun, exciting? I think it's fun. I think it's neat. I think it's exciting. Why are parts purple and not purple? Does anybody have any guesses or ideas or thoughts?
I think it's just so we can see the difference.
It does. It's almost so we can see the difference. Every chromosome will stain in this pattern shown here. For whatever reason, some parts stain more heavily than other parts. And it's usually what's called GC banding. To just show you different regions of the chromosome. It's not heterochromatin, euchromatin. Don't equate purple with euchromatin or heterochromatin. But it's just showing you different regions of the chromosome. And mostly it's there for locationally speaking. Where are things? Now when it comes to where are things, there is an important thing to think about with each of these chromosomes. How are they put into the nucleus? And more importantly, how does a cell know where to go to find the DNA that it needs to find to make it the cell that it is? Liver cells don't use the same DNA that heart cells do. Otherwise they'd be the same type of cell. So different things, different genes, different proteins needed for different functions. How does the cell know to go here to find this DNA or here to find this DNA? I don't know, but there is an organization to the the nucleus. And chromosomes have basically their spots. It's like Sheldon. Everybody has their spot. And every cell, the DNA should be organized exactly the same. And each of these different colors is supposed to represent a different chromosome. Sometimes it's kind of hard to see all the different colors that are in there. But what we do see when we look at the chromosome, there is an organizational format that's important, and it has to do with the euchromatin areas versus the heterochromatin areas. And generally speaking, the DNA that we use most often, the DNA that's most heavily transcribed or expressed, will be in the center of this sphere. So the euchromatin, by definition, should be in the middle of this three-dimensional ball. And somewhere on the outside of the nucleus, or the exterior parts, out here, would be where we find the heterochromatin DNA in the middle. The stuff you need most is readily accessible, kind of in the middle. Stuff you need a whole lot of on an everyday basis, sort of on the peripheral of your living space. Think about your apartment or your room or your house. Where do you put the things you use the most often? In a way out of the way place? Or in a place that's easily gotten to? The things you don't need very often, put in the attic, put in a storage box, underneath your bed, or in the garage, or at your parents' house, because you don't have any space. So that's just a general organization sort of concept for the nucleus. In this three-dimensional sphere, the parts that we need the whole lot are typically in the center part, easily accessible. The things we don't need a whole lot are more on the outside of the nucleus. But still, I think it's pretty amazing that DNA polymerase or RNA polymerase can go in here and take the right piece of DNA to make the right RNA so we can make the right protein. Is it like really tight and packed, or is there spaces in there? I don't really have the answer to that. It's kind of hard to see if there's spaces in here, but maybe you can imagine there's space in this wall where things can move around and it's not like a solid sphere. But every location has a unique area inside the nucleus. This slide won't take you long, so we'll go ahead and throw it in there. Get it out of the way. Every chromosome needs its centromere so that it can be moved around properly during cell division, like mitosis and meiosis. But sometimes problems happen where centromeres are lost or duplicated. Right? It's going to create an issue. The acentric chromosome has no centromere, which means it can't get moved around during cell division. So some cells are going to get extra DNA and one cell is going to get less DNA. That's going to be a problem. If you're missing an entire chromosome, you're missing a lot of what makes you a human. Can you imagine why two centromeres might be problematic? Wouldn't two places make movement faster? Could be uneven, but also could be in the opposite direction. So if you have things pulling this thing apart, then you get broken pieces of DNA going in different directions. So two could create problems because of the movement and never know where they're going to go. Now, what makes the chromosomes without a centromere or with two are some funny little effects that we call crossing over replication, which are fun things to think about as we go forward. You might have heard of the term crossing over when talking about meiosis. The recombination is like crossing over, but slightly different. We will get telomeres on Wednesday, I guess, because